You are watching single-cell multiome analysis tutorial series with SciDAP. In this quick video, we will review the output of our FASTQ download pipeline. You could click on one of the samples that finished running. SciDAP summarizes all the information about the sample you uploaded, the project number it belongs to, the type of the data, and some statistics of the SRR run. And here it provides statistics of every FASTQ file that data been split into. In this case, we see 4 FASTQ files. You could even see how reads look like. For the next step, we would not need all of the FASTQ files. Some of them are 10x indices that were necessary for the run, but useless for the downstream analysis. They could be distinguished by read length. On 10x Genomics website, it is shown that sample index I7, that has read lengths of 8 base pairs, was used for demultiplexing. So we will not need this file, but we will need other 3. Let's see. Similarly, for RNA-Seq, there are only 2 files that are needed as input for SciDAP. This is read 1 and read 2. And the third file will be index I7 that we don't need. The length of it is 10 base pairs, so remember this. Let's see. The first one has read length 8. Yes, this is the file that we will not use for the downstream analysis, but the other 3 we will need for the next step, which called cell ranger count. However, if you need all of the files for your custom analysis, you could easily find them in this tab called Files and download not just the FASTQ files that are compressed in GZ format, but also metadata or log files. And in the next video, I will show you how to start analyzing your data using the pipeline called cell ranger count.